This video abstract is AI generated. How can a small change in protonation reorganize an RNA structure? This manuscript focuses on the U6 internal stem loop, or U6ISL, a highly conserved RNA element of the spliceosome. The internal loop of U6ISL contains three key nucleotides, C67, A79, and U80. Previous work showed that this region behaves as a pH-sensitive conformational switch. At higher pH, U80 is mostly stacked in the helix, and this state favors binding of an essential magnesium ion. At lower pH, A79 becomes protonated, a C67 A79 plus wobble pair forms, and U80 flips out of the helix. The biological question was clear, but the mechanism was not. What structural pathway connects these two states? And why does protonation of a single adenine shift the RNA toward a flipped-out uracil conformation to isolate the high pH endpoint? The authors solved the NMR structure of the U6ISL at pH 8.0. This structure showed that when A79 is unprotonated, C67 pairs with U80, leaving A79 unpaired. This C67U80 pair explains why U80 prefers to remain stacked in the helix at higher pH. Molecular dynamics then revealed that the high pH and low pH states have very different internal loop behavior. In the high pH structure, the C67U80 pair breathes. It breaks and reforms during the simulation. These breathing events allow transient contacts between C67 and A79. In contrast, when A79 is protonated, a stable C67 A79 plus wobble pair forms. Once this pair is stabilized, U80 becomes unpaired and flexible. The next challenge was to describe the transition itself. Because base flipping is slower than ordinary molecular dynamics can easily sample, the authors used nudged elastic band simulations to calculate a minimum energy path between the stacked and flipped-out states. The path was represented by a series of images connecting the two endpoint structures, and the orientation of U80 was tracked with a pseudodihedral angle. The result was a clear mechanistic picture. When A79 is protonated, the transition begins with base pair switching. The C67U80 pair breaks and the C67 A79 plus wobble pair forms. Then U80 flips out through the minor groove. When A79 is not protonated, U80 can still explore flipping motions, but the stacked C67U80 state remains the lowest energy state. The final model is that protonation, base pair rearrangement, and base flipping are coupled. A79 protonation reshapes the internal loop energy landscape, stabilizes the C67 A79 plus wobble pair, releases U80 from the helix, and promotes minor groove base flipping. This work shows that small shifts in nucleobase pKa can reorganize RNA structure and tune the functional landscape of the spliceosome. To know more, read our work online.